Assessment questions 12 through 16 are going to look at mutations. So you're going to be making mutations in the gene that you described in question number 1. A point mutation is where there is an addition, a deletion, or a substitution of a few bases. So a few bases, adenine, guanine, cytosine, or thymine, are going to be changed in the DNA. Sometimes this change is going to result in a change of the phenotype. Sometimes it doesn't. There will always be a change in the genotype when there is a mutation, but not always in the phenotype. A silent mutation involves the changing of a base, but it doesn't change the amino acid, and hence it's called a silent mutation because it causes a change in the genotype, but not in the phenotype. So there's not going to be any impact on the protein that's synthesized from the messenger RNA that was transcribed from the gene. Substitution mutations can result in missense. A missense mutation changes a single amino acid, whereas a nonsense mutation changes a normal codon into a stop codon. A number of things can cause mutations. They're not only chemical mutagens, but there are also physical mutagens. Point mutations can be caused by nitrogen-base analogs, and those are molecules that look like a nitrogenous base. 2-aminopurine is one, so it substitutes in for purines. It's used in cancer treatment. AZT is azidothymidine, which substitutes in for thymine, and it's used to treat HIV. Cytarabine and 6-mercaptopurine are going to be used to treat cancer, and Zovirax is a nitrogen-based analog that is used to treat herpes infections. Caffeine is also a nitrogen-based analog, and we know that caffeine is a mutagen, and it is the kind of thing that you have to limit when you are pregnant. These nitrogenous-based analogs are going to compete with the natural bases, and when they are incorporated into the DNA, they actually distort the helix because they aren't the right size, and that leads to mistakes in base pairing. Alkylating agents such as nitrogen mustard, which was used as a gas in World War I, busulfam, and cyclophosphamide are going to add an alkyl group, which is a single bonded carbon and a hydrogen, to a base, and this alters its binding capabilities, altering its means of making hydrogen bonds. So point mutations are going to be a single change in the DNA, perhaps one or two. We don't see a great number of bases being changed with these mutagens. Frameshift mutations are going to alter the reading frame. Now, as you've learned so far in this chapter, the reading frame is in groups of 3. So you have your codons. First you have your triplets, and the triplets are going to code for codons, and all of this is in groups of 3. The ribosome translocates in units of 3. If a base is removed, you've now shifted the reading frame, or alternately, if a base is added, the reading frame has been shifted. Some things that can cause frameshift mutations are acroflavine and proflavine. These are acridine dyes. Ethidium bromide and quinacrine. And quinacrine and ethidium bromide are molecules that intercalate in the DNA molecules. In other words, they insert in between the nitrogenous bases. And when they are there, then they are going to prohibit transcription and translation. Transcription will be inhibited, and so will replication, simply because these molecules are acting like blocks or boulders, prohibiting DNA polymerase or RNA polymerase from either replicating or transcribing, whatever their job is at that time. So a frameshift mutation can be caused by ethidium bromide, which is commonly used to stain DNA in processes where we want to visualize it in genetic engineering or biotechnology. So assessment question number 12, you are going to create a silent mutation in the gene. You want to show this mutation in red. So that red base is going to be shown in the DNA, right? Then you're also going to show the change in the messenger RNA codon in red. But this is not, remember, a silent mutation is not going to change the amino acid. So you're going to copy the gene from question number 1 and change one base that changes the genotype, but not the phenotype. Change the font color to red for that base pair in DNA, and change the font color to red for the base that changes in messenger RNA. So the change in the codon, not the entire codon, but just in red, the one that is changed. So how are you going to figure out which one to do? Well, if you take a look at the genetic code, you can see in the genetic code that UUU codes for phenylalanine, and so does UUC. So if there is a change in the U and it is changed instead into C, right? So this would be a change from thymine to guanine. Sorry about that. Change from thymine to guanine in the DNA, then this is going to result in a change in the messenger RNA from UUU to UUC, back here, okay, to UUC, and this is still going to code for phenylalanine. Because the DNA code is degenerate, the codons are degenerate, you can see here that C and U are the same here, so any change in this third position will still result in the same amino acid being incorporated. So you can see that this type of mutation, a silent mutation, is not at all devastating to the cell. It makes absolutely no difference in the phenotype. Okay, so I'm going to show that in red, and then you'll show the change in the messenger RNA codon in red as well. Okay. A missense mutation now is going to change both the genotype and the phenotype. So again, you're going to copy the gene from question number 1, and as I mentioned in class, the easiest thing to do here is just copy-paste the whole thing in rather than trying to type in everything again. And then you're going to change a base in DNA that will change the messenger RNA to code to a different amino acid. So this is a genotypic and a phenotypic change. Here you're going to indicate the mutation in red, like you did in the previous question, indicate the change in the messenger RNA in red as well, and then indicate the change in the amino acid in red also. Remember, you don't want to carry the mutation for the previous question over to this question, so you're always going to be starting back with the gene from number 1. So again, when you take a look at the genetic code, you can easily see that if you change the second position or mutate the second position or the first position, that is definitely going to change the amino acid. Whereas if you change that last position, you can see in this box here, changing that last position will not change the amino acid. So shoot for changing either the first or the second of those bases in the triplets, or in the codon, excuse me, and that is going to cause a change in the phenotype. So that was question number 13, your missense mutation. A missense mutation is going to change the genotype and the phenotype. Now you're going to create a nonsense mutation in the gene. The nonsense mutation changes a regular codon into a stop codon. You may have to go back. You can actually change 2 nitrogenous bases when you do this, and not just 1, but you can't change 3. So if necessary, you may have to go back and go all the way to the beginning in question number 1 to make a change, so you can get this change for question number 14. Remember, if you do make a change to question number 1, you are going to have to go back and make all of the changes to everything all the way through. Okay. So the nonsense mutation is going to change your regular codon into a stop codon. So you see we've got UAA, UAG, and UGA. So hopefully somewhere in there you either had a tryptophan or a tyrosine that was encoded in your primary sequence of your amino acids, of your protein, and hopefully you can just simply change it. But what you are now going to do is show the changes in DNA in red, and then you are going to show the changes in the RNA in red. Okay, so remember here that just because there is a stop codon that shows up here in the middle of your gene, that doesn't mean that these bases here stop. Go back and remember what a stop codon means. Stop codon does not stop transcription, it only stops translation, right? So this is messenger RNA on the bottom. The messenger RNA is just transcribed from the DNA. So you are now going to have a stop codon, but it's going to be somewhere earlier rather than in this position where it was before. It's going to be somewhere in here. Okay, so it may be necessary to mutate 2 bases to get to that point. Don't carry over the mutation from the previous question. Again, use that gene from question number 1 in its entirety. So question number 14 is making the nonsense mutation. Right? Number 15 is going to make a frame shift mutation. Remember, a frame shift mutation causes a change in the reading frame either due to an addition of a base or a deletion of the base. You're going to do a base deletion. So you are going to delete some base in the DNA, pointer there. So you're going to get rid of one base. So you're going to get rid of a base pair, actually, and this is going to impact the messenger RNA because you've now shifted the reading frame. So leave one set of boxes blank in the DNA to show the deletion. Okay. You will lose the original stop codon that will be in the messenger RNA, but you may gain another. It depends upon the sequence that you decided in question number 1. Change the font color to red for all of the amino acids that are changed as a result of the mutation. Okay. So you're going to indicate the change in the primary sequence. That's the amino acids that are in these bottom boxes. So the amino acids in the bottom boxes are going to be affected as a result of the frame shift mutation, and those that are affected change to red. Change your font color to red. Okay. Assessment question number 16, you're going to name an agent that causes frame shift mutations. So go back a couple of slides and name an agent simply from the slides and explain the effect that this frame shift mutation has on the translation of your protein and translation in general. So remember to consider the role of the stop codon when answering this question. The stop codon is going to affect translation, but not transcription. We'd also like to look at what ultraviolet light does to DNA. Ultraviolet light is a very powerful mutagen, and it kills microorganisms, although microorganisms have evolved mechanisms for getting around this. Ultraviolet light induces dimerization between adjacent pyrimidines. So dimerization is a formation of bonds between 2 pyrimidines, hence a dimer. We're going to see adjacent pyrimidines, that means next to one another on a strand of DNA, getting abnormal bonds. We see this between adjacent cytosines, between a cytosine and a thymine, or a thymine and a cytosine, and also between 2 thymines on the DNA. This dimerization is going to cause local distortions in the helix. It weakens the interaction with the complementary bases, and it kinks the backbone slightly. The formation of this 4-carbon ring between the pyrimidines makes it difficult for DNA replication enzymes to determine what base pairs should be across from the fused pyrimidines, and a copying mistake like this can change how the DNA encodes a protein, and as a result, an abnormal protein can be synthesized if the transcript is even made, even if the messenger RNA may not be made. Okay, so here you can see we have the nitrogenous bases in DNA, so there'd be the 5' and the 3' strand here. And if ultraviolet light strikes these strands, then the adjacent thymines or adjacent pyrimidines are going to be affected. Both strands of the DNA will be affected. Now, in this example where we see there are no others that would be affected, only these two thymines here, but keep in mind that both strands, the 5' to 3' and the 3' to 5' strand, will be affected by ultraviolet light. Any 2 thymines that are next to each other, and to a lesser extent, any two cytosines that are next to one another, or a cytosine and thymine that are next to one another. It primarily causes thymine dimerizations, but all pyrimidines can be affected. So here you can see abnormal bonding that is happening between these thymines, and this is going to affect transcription and translation. So now you're going to go back to question number 1, and you are going to imagine irradiating this DNA with ultraviolet light. You're going to draw connecting lines between the affected bases. You can do this by hand, or sometimes students will put an asterisk next to the letter that shows the abnormal bond that's being formed. So you can do this with a pen. Any place you have two C's next to one another, or two T's next to one another, or a C or a T or a C. And remember, both strands are going to be affected. Just draw a line in between these, showing how DNA is going to be affected by ultraviolet light. Now, bacteria, as I mentioned before, are quite efficient at repairing damaged DNA. We do use ultraviolet light in order to kill kill bacteria in water specimens or water treatment plants. So ultraviolet light can be used for that. Ultraviolet light is used in operating rooms. At the end of the day, they will turn on a bank of UV lights, and that's going to kill bacteria that come into contact with the UV light. And also the biological hood that you'll be using in the fungus lab when we finish that up, that also has ultraviolet light in order to kill any bacteria or fungi that will be left in the biological hood when we're done. So there are two types of repair that bacteria can use. One is called excision repair, and this involves a protein called a UVR, or an ultraviolet repair protein. It removes the damaged segment, so it is an enzyme. Then DNA polymerase I, an enzyme that you met before in the beginning of this assessment, fills in the gap, and then a ligase seals it. Eukaryotes can also repair damage, and this happens with your skin cells. When you get too much ultraviolet light exposure, your skin cells can repair the damage using excision repair. Another type is called photoreactivation, and eukaryotes can't do this. This is when an enzyme called DNA photolyase attaches to the bond between the pyrimidines, and this happens in the dark. When photons or light strike the enzyme, the bonds are broken. In this case, no gap filling is necessary, no ligase is necessary to fix the problem. There's no gap filling necessary. So let's take a look at a video on these two processes.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of two UVRA molecules and one UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the two UVRA proteins are released. UVRC then binds to the site and makes two cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVR B, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
So notice in that video that there was much more of an explanation, and please include that information on question number 18. Now let's take a look at the video that explains photoreactivation.
Hello friends, this is from easybiologyclass.com. In this tutorial, we will discuss about the photoreactivation method of DNA repair. As the name suggests, it is a light-dependent DNA repair mechanism which rectifies UV-induced damages of DNA. This is the part 2 of the tutorial series on DNA repair mechanisms. In the first part, we have discussed the introductory portions of DNA repair, such as DNA damaging agents and different types of structural lesions in the DNA induced by these agents. Those who are not aware of these topics may please watch the part 1 of this tutorial and can return to this since it is the continuation of the part 1 tutorial. In this session, we will discuss about what is photoreactivation, the mechanism of thymine dimer formation by UV light, a brief note on the discovery of photoreactivation mechanism, then the details of enzyme involved in photoreactivation process, that is photolyase enzyme, and finally a detailed molecular mechanism of photoreactivation process. Okay, let's see what is photoreactivation. Photoreactivation is a type of DNA repair mechanism present in both prokaryotes and eukaryotes. It is a recovery of ultraviolet irradiated damages of DNA by visible light. As the name suggests, it is a light-dependent process. It is a DNA repair method by which cell recovers its DNA after UV exposure. The UV light is lethal to cellular DNA since it induces structural lesions in the DNA by the formation of pyrimidine dimers. Photoreactivation process removes the pyrimidine dimers without altering other nucleotides in the DNA by using energy obtained from the visible light. Photoreactivation is the first discovered DNA repair mechanism in the cell. Major credits for the discovery of photoreactivation goes to Professor Aziz Sanger, for which he was shared the 2015 Nobel Prize in Chemistry along with Thomas Lindahl and Paul Modrich. As we mentioned, the photoreactivation process removes pyrimidine dimers. Before going to the photoreactivation process, let's see how UV radiation induces pyrimidine dimers in the DNA. As we have already discussed in the part 1 of this tutorial, exposure of DNA with ultraviolet light causes pyrimidine dimer formation. Among the different types of pyrimidine dimers, the thymine dimer is the most common one. Here, two thymine residues adjacently situated in the DNA strands are covalently bonded to each other and thus cause a structural lesion in the DNA. Thymine dimer is also called the cyclobutane photodimer or CPD because it structurally resembles the cyclobutane nuclease. As you can see in this image, this is a cyclobutane nuclease and this is a thymine dimer. One thymine here and another here, both connected by two covalent bonds. This is a normal DNA duplex with the usual Watson-Crick base pairing. Here you can see two thymine residues situated adjacently to each other. When this DNA is exposed to UV light, the hydrogen bonds between the two strands breaks and the two covalent bonds are produced between the thymine residues. Then let's see how the covalent connection is formed between thymine residues by the UV light. These are the two thymine residues of the DNA. When UV light hits this DNA, these double bonds which present between C5 and C6 of both thymine breaks. So in order to balance the valency of carbon, the electrons are shared between C5 of one thymine with the C5 of adjacent thymine, and also the C6 of this thymine bonded with the C6 of adjacent thymine. As a result, two covalent bonds are formed between the thymine residues, which we call as thymine dimer. Thymine dimer induces a bulge in the DNA, and as a result of this, the normal conformational symmetry of DNA is lost with this lesion. Okay, then Let's see a short history about the discovery of photoreactivation process. In 1920s, Hermann Muller discovered that X-rays can induce mutations in the cell. Later in 1940s, Albert Keller, with his simple experiments, discovered that UV-induced cellular damages can be recovered by treating the cells with a visible light. He called this process as photoreactivation, since it is a light-dependent reactivation or recovery of cells. Then a few years later, Renato Delbecco suggested that photoreactivation may be an enzymatic process dependent on visible light. However, he was not able to prove this experimentally. Immediately after this, Stan Rupert proved Delbecco's theory of enzymatic photoreactivation, and he named the enzyme as photolyase for the first time. The discovery of photolyase was very remarkable in the scientific community because for the first time it proved the existence of such an enzymatic DNA repair mechanism in the cell. Then in 1978, Professor Sanger cloned and purified the photolyase enzyme of E. coli. Sanger was at the time a PhD student with Professor Stan Rupert, but instead of continuing to characterize the photolyase, Sanger wrote his PhD dissertation and graduated. After 4 years, that in 1983, Sanger returned to his original work of photolyase enzyme, and he characterized the enzyme, and also he explained the detailed molecular mechanism of photoreactivation process by visible light. Then let's see the molecular mechanism of photoreactivation. As we said, photoreactivation by the enzyme photolyase is a light-dependent process. Photolyase is a flavor protein and it contains two chromophores which absorb light energy from visible light. The chromophores act as a catalytic cofactors. The two chromophores of photolyase are a fully reduced flavin adenine dinucleotide or FADH negative and a folate group. The fully reduced FADH- is very essential for the catalytic property of the enzyme. Photolyase enzyme absorb light and get activated. The light-activated photolyase enzyme splits the thymine dimer by a local free radical mechanism by a transient electron transfer method. First, the photolyase enzyme recognizes the thymine dimer lesion in the DNA and binds to it. The enzyme then absorbs visible light at 200 to 500 nm, that is near blue region. As a result of the light absorption, the FAD negative cofactor gets excited and elevated to a higher energy state. Excited FADH negative transfer an electron to the thymine dimer. This electron interacts with the thymine dimer, and as a result of this interaction, a charged separated radical pair is produced. The unstable radical pair suddenly splits, and thus the connecting two covalent bonds between the thymine breaks. Once the connecting bonds breaks and the thymine residues are recovered, the electron is returned to the FADH-. Then the photolyase enzyme leaves the DNA and the normal conformation symmetry of DNA is regained. This is how photolyase enzyme removes the pyrimidine dimer from the DNA in a light-dependent fashion. Finally, let's see the significance of photoreactivation. As we mentioned, the photoreactivation was the first DNA repair mechanism identified in the cell. It is a direct reversal method of DNA repair in the cell. Here, the enzyme do not require the assistance of any template strand. More than that, there is no removal of nucleotides are involved in photoreactivation process. Photoreactivation is not a universal DNA repair mechanism. Photoreactivation mechanism is present in bacteria, archaebacteria, plants, and in many animals. This method of DNA repair is completely absent in placental animals, including human. Placental animals, including human beings, uses nucleotide excision repair or NER to remove the pyrimidine dimers from the DNA. Okay, this is all about the photoreactivation DNA repair mechanism. In the next tutorial, we will see the molecular mechanism of base excision repair and nucleotide excision repair.
All right, so when you are answering the question, number 18, how the organism might repair the DNA damage done by irradiation, make sure you watch the videos and include the detail that is found in the videos. So this is 18, describe how the organism might repair the damage. Make sure you are using your own words, so don't copy the information directly from the presentations, from the YouTube presentations, but instead put this in your own words. Okay. The next set of slides, we're going to take a look at intermicrobial gene transfer.